Vectors for use in Saccharomyces cerevisiae are yeast. In order to clone heterologous or foreign DNA into the yeast cell, sometimes it is important that it is ligated within a suitable vector, and those vectors must be able to first replicate in other host organisms like the E. coli. कि इसमें हम जो फॉरेन डीएनए है या हमारा जो जीन ऑफ इंटरेस्ट है, इसको हम पहले किसी प्लाज्मिड में लाइगेट करके तो इकोलाई में हम इसको रेप्लिकेट करवाते हैं. इसका एडवांटेज ये होता है कि जो हेटेरोलोगस डीएनए है या फॉरेन डीएनए जो है आर डीएनए फ्रॉम अनदर सोर्स दैट इज अदर देन यीस्ट, इट कैन बी प्रोपेगेटेड विद इन द इकोलाई. तो इस तरह से हमारे पास हेटेरोलोगस डीएनए की बहुत सारी कॉपीज जो हैं वो आ जाती हैं. फिर इन कॉपीज को इंट्रोड्यूस करवाया जाता है यीस्ट के अंदर. नो वैरायटी ऑफ प्लाज्मिड्स हैव बीन डेवलप्ड दैट हैव द एबिलिटी टू रेप्लिकेट इन दीज टू होस्ट्स. लाइक इन ई. coli, they are first propagated and later on they can be introduced into yeast cells. Here, the plasmids that are used, they must have some desirable properties as a general rule. If you remember that they must have multiple cloning sites, that is target sites for different type of restriction enzymes, ताकि इसको एक खास restriction enzyme से digest किया जाए, and then foreign DNA can be ligated. And of course, what is the size of the foreign DNA can be cloned? It is also very important. And some selectable markers that after transformation, they can be used for the selection of transformants. And here, variety of plasmids are available for this purpose, but we can quote a few examples. Like yeast episomal plasmids, we can simply abbreviate them as YEPs. And in addition to that, yeast replicating plasmids or YRPs, they can also be used. But the most suitable one for the cloning in yeast, it is the next type, that is YCPs, and it can be simply called as yeast centromere plasmids. Now, what is the reason? We are going to discuss it in next topic. And the most famous one that we have discussed a number of times, it is the yeast artificial chromosomes, and simply we can call it as the YACs, and it is very much favored for cloning with yeast. तो हमने काफ़ी दफ़ा डिस्कस किया है कि इसकी जो डिज़ायरेबल प्रॉपर्टीज़ हैं, इनमें एक ये भी है कि अगर हम इसको बैक्टीरियल प्लाज्मिड से कंपेयर करें तो इसमें जो फॉरेन डीएनए का जो फ्रेगमेंट है जो क्लोन हो सकता है, उसका साइज मोर देन 1 मेगाबेस तक भी हो सकता है. बट द मोस्ट इम्पोर्टेंट or the example that we are going to focus here, it is the yeast episomal plasmids or YEPs. And this plasmid, it has been developed by using origin of replication from another artificial bacterial plasmid that is called as the PBR322. And even the selectable marker like the ampicillin resistance gene has been taken from this plasmid, and all these properties have been combined with the tetracycline resistance gene or the region of replication with the yeast plasmid, and this example we are going to discuss in the next diagram. So this is the example of typical yeast episomal plasmid, and it is called as simply YEP24. So this is the example that we are discussing here, and it contains region of replication from PBR322, then ampicillin resistance gene that can be used as a selectable marker, and then URA3 gene, it contributing another region of replication. So two region of replication, one from bacterium, another from yeast. It is helping them to multiply in these two diverse host organisms. Another selectable marker, it is tetracycline resistance. And some genes we are also taking from yeast plasmid, like 2 micrometer plasmid. It is an example from the yeast. And variety of cloning sites or restriction sites for different restriction enzymes, they are also present along the whole circle of this yeast episomal plasmid.